what approaches are necessary to get at dysfunction within patient networks that can allow us to personalize therapies for the unique characteristics of the tumors. And the holy grail here, as Lee mentioned, is to build a capability to predict, prevent, and cure cancers and any complex disease. And this builds on the basic assumption that we can find within these large amounts of data that we can discover signatures, molecular signatures that can allow us to stratify patients into quantitatively into subtypes of disease and use that information to find dysfunction within the gene networks to personalize therapies. So in a conceptual framework, this means that we can use our understanding of the disease and molecular signatures to differentiate a healthy state from a disease state and find the unique features across different types of data, be it the genome sequence, the expression of genes, the interactions among proteins and modifications of proteins and so forth, that uniquely characterize the healthy state versus a disease state. If you're able to do that, we can then quantitatively diagnose a disease, but it doesn't really help you predict pre-symptomatically if someone's risk for a disease has increased or decreased. To be able to do that, we have to collect data that allows us to look at transitions from a healthy state to a disease state. And if you have that type of data, you can then begin to make predictions, even with just correlates. So you can find features that correlate with the transition and use that information to generate a risk assessment of someone's likelihood of getting a cancer. But correlations do not equate to causation, and I'll get to that in a minute. So that information does not give you the necessary information required to block the transition or even prevent or reverse a disease once a person has it. So how do you go about finding these signatures so you can not just predict, but you can also do rational intervention to prevent transition or promote cures? And here's the approach. The assumption here is, given a cohort of patients, if the cohort is well designed and large enough, then you can assume that within that cohort are subset of patients that share a similar dysfunction in the disease. The challenge is to mine through different data sets and do in a data-driven manner, the stratification of patients into subgroups, and then to rank order them in a data-driven manner based on severity or progression stage of the cancer, and then exclude patients who have a different indication. If you're able to get signatures like this, then the question is, what was the underlying dysfunction that was responsible for generating these signatures in these patients? What is the common mechanism of dysfunction? And this becomes extremely challenging because when you have a dysfunction in a network, and as Lee mentioned in his keynote, we are systems of systems. We have networks of genes and cells and tissues and so forth. So you have dysfunction in any one element. That dysfunction has consequences that propagates throughout the network. So when you make measurements using these powerful technologies, what you're measuring are not just the primary dysfunction, but consequences that may be 1, 2, 3 steps or even more removed from the primary dysfunction. And then you end up with these large data sets, very complex data sets. And you've seen some examples from Ralph. These are very complicated sets of information, and you can have many different kinds of information going from the genome sequence, the expression of genes at the RNA level, at the protein level, protein interactions, protein modifications. So you are piling hay over the stack and trying to find a needle within that haystack. And the general approach that people take is to find correlations across features. So you see a change in a feature, and you look for a correlated change in another feature. And then you infer from that some form of association between those two features. Now, when you deal with molecular system-wide data, the features you're dealing with are typically cryptic chemical names, gene names, molecules, and so forth. And often, when you draw an edge between these features, you feel good that you found a correlation, but the question is, does that mean anything, right? Now, there are other examples of what could go wrong when you have very large amounts of data, and there are websites There's one particular website which I like that is devoted to these coincidental correlations in very large data sets. And just to make this point that if we knew what the features meant, what kind of information we might be dealing with, I have some examples here. So you have correlation between people who drowned after falling out of a fishing boat and marriage rate in Kentucky. Doesn't make any sense, but look at the data. It's beautifully correlated, right? And these are data from CDC, and these are reputed sources. Here's another example. You have a correlation between the age of Miss America and murders by steam, hot vapors, and hot objects. Okay, and here's the third one, where you have per capita cheese consumption correlating with number of people who died by becoming entangled in their bed sheets. Okay, it sounds ridiculous because we know what these features are, and we know they don't make sense in terms of being correlated with each other. This is coincidental. So this is what happens when you have very large amounts of data. The likelihood of finding correlations by chance is very high. It can be very misleading. So we have to figure out more principled ways that are based on biological mechanisms and prior knowledge to guide this discovery process. So I'll explain how this could be done. The goal is to go from correlation to mechanism and find causation in the process as well. So you can then say, because of a dysfunction here, there's a consequence over here. So if I block this, I can reverse this consequence or block the consequence. So how do you do that? So a general schematic for this would be if you had a cell line or an organism that you can perturb in controlled ways, you could expose it to slight changes in temperature, some drugs, pH changes, and so forth, nutritional changes, and then measure outputs of that system. You can look at how gene expression changes, and you can collect a large compendium of data. And then the goal is to figure out what was the architecture of interactions among components of that system that gave rise to that output. Now, the architecture of interactions we're dealing with here are architecture of gene-gene interactions at a molecular level, at protein-protein level, at protein-DNA level, RNA-protein level, RNA-RNA. So we have known mechanisms of how these interactions could occur. So we can impose constraints in this discovery process. It's a reverse engineering process that can then elucidate from the output the structure or the topology of the network of interactions within the system that was responsible for sensing the changes you imposed and process that information into the output you measured. And if you do it well, then you should have predictive power, meaning you should be able to make predictions of what would happen if you perturb that network on the output of the system. And you can test that in cell lines. But you can't do this in patients. You can't bring a patient in and perturb them. So what you have to deal with are patient cohort data. So you have cross-sectional data, one measurement from one tumor biopsy of one patient, and many such measurements. And all of these samples are mixtures of cells, and the patients have different histories, and there's a lot of complexity here. So we have to navigate through this complexity and then treat these patients as snapshots of the disease captured at different time points. Okay? But you have to make sure you are grouping the patients into meaningful subpopulations. So I won't go into the details of these algorithms, but I want to give you a flavor of how these algorithms work. So here's one algorithm that does multi-clustering. So the goal of this algorithm is to navigate through these large data sets and find genes that have similar changes in expression across subsets of patients. But we don't want coincidental correlations for reasons I described earlier. We want correlations that can be supported by evidence of some mechanism responsible for that correlated change. So some mechanism of regulation, a protein binding to the regulatory region of a given gene, or a small RNA binding to the 3' untranslated region of its transcript that is shared among genes that are correlated to be able to draw or infer that this correlation makes sense. So we can also input information regarding coevolution of genes, interactions amongst proteins, protein-DNA interactions as priors in the network shown on the left below here. So this is a movie. You'll see this algorithm just navigates to this data space and starts grouping genes, throwing genes out, throwing patients in, throwing patients out, finding these signatures de novo, using that information to find additional patients that should be grouped into this and genes that needs to be grouped into this cluster. And then you converge at some cluster that looks like this, which has information regarding which patients share a signature of similar gene expression changes, and evidence that that correlated expression is because of putative regulatory mechanisms they share, and because of some mechanistic interaction amongst those elements. Okay? And once you have that signature, then you can ask, okay, what was the topology of interactions that gave rise to that signature? And there's many different algorithms that can be used to do that, to map mutations onto the network, to map proteins onto the network, to map RNAs onto the network. And these are players that are modulating the expression of genes. And if we can figure out what these players are, then we're beginning to get to the mechanistic dysfunction. So I'll show you a couple of algorithms. There's one approach you can use that takes advantage of regression analysis. So if you had information on activity of these regulators, then you can use that to find those regulators that change in a temporally preceding manner before you observe change in expression of genes in a given cluster. Okay? And the way you do that in patients is a little tricky, but if you're able to do that, you can make predictions, because this is a quantitative equation that predicts how changes in these elements are imposing consequences on changes in expression of genes within these clusters. And you can go and do experiments to test in a new cohort of patients whether you have made inferred an accurate model. And we've shown in microbial systems, where we originally developed this approach, that you can make predictions for genes across the entire genome of a simple organism. Okay, the question is, can you do it with a human system with all the complexity that we have to deal with? For the approach in human system, we have to take into account the complexity of how gene regulation occurs. So you can have a gene being regulated by some protein that is bound tens of thousands of base pairs away from that gene. Okay, so it's very complicated. So some of the techniques that we had originally developed were not sufficient. So we developed this new technique where when you know the binding sequence of a transcription factor in the genome, you can scan the entire genome for matches to that recognition sequence. Now, these sequences tend to be short, about 6 to 8 nucleotides, and the human genome is 6 billion bases. So the likelihood of finding these short sequences is near random. So you need to find those matches that are most likely to be functional. So we can then take another data set, and this is a data set where a group of researchers across the US, across the world actually, measured accessibility of the human chromatin by little enzymes or by DNA cutting enzyme. And if the enzyme is able to cut the DNA, then you know that the chromatin was accessible. So if the sequence is in a location where the enzyme could access it, that means the protein that binds it might also be able to access it. So there are 3 locations where we found matches to this protein sequence binding, potential binding, and found only 2 to the left are in locations where the chromatin is accessible in one of 200 different human cell types. Then we can zoom in even closer into that data and ask, in that location, in those 2 regions that were accessible, is there evidence that a protein was bound to that region in the genome in any of those cell types? And that's called a footprint. And so we find that of the 2 that were accessible, only 1 was is associated with a footprint. So we can then infer that of the 3 matches, the protein could bind only to 1 of those locations. So we can create a big database like this for all the transcription factors for which we have this information. So it's thousands of transcription factors, and confirm that in other ways as well. So I'll skip this. We can do the same for microRNA regulation. So microRNAs are the small RNA molecules that bind to the 3' region. So the region of the RNA that does not translate into a protein has regulatory information, and it has 6 to 8 nucleotide sequences that can be bound by small RNAs. And we can find those sequences across these correlated genes, align them, calculate the frequency of nucleotides at each position, and use that frequency to generate a hidden Markov model, and use the hidden Markov model to find which RNA was bound there. So there's many different ways you can get at the regulators of these clusters. And again, I'll skip that. But what we've done is we've taken a series of algorithms like this and strung them together in ways that allow us to assess the statistics of these discoveries using scoring functions that can make sure that you are finding meaningful information and you're throwing away coincidental correlations. And when you put patient data through this pipeline, you can generate networks that can then allow you to map mutations in the tumors of patients to specific regulators, transcription factors or microRNAs, who are predicted to control specific clusters of genes. And because we know what those genes are, we can, through gene ontology mapping, predict what processes they're associated with. So we can then say this mutation is causing this perturbation of, in the processes known to be associated with cancers. And so I'll quickly show you an application to glioblastoma, where we took about 422 patient tumor biopsies. We took disparate data from these tissues, we processed it through this pipeline, and discovered that there are about 1,830 to be precise, clusters of genes that have these signatures that I showed earlier. Of these 1,800, we found that there were about 500 that could be associated with some cancer process. And among that, we reduced it to a set of 200 odd clusters that had both a cancer-related process that was perturbed and was associated with clinical outcome like survival. So these we call disease-relevant signatures. And for these clusters, we identified the regulators and mapped mutations onto the regulators and showed that there's high degree of overlap between causal information propagation through that network and mechanistic information propagation. And I'll show you some examples regarding what that means. So here's one signature as an example. So here you have a signature of about 16 genes or so. And these genes, the median expression of these genes is used to stratify patients. So every dot along this trajectory is a patient tumor biopsy. And they're rank ordered based on the median expression level of these genes. And about 20 patients were excluded because the variation in expression of these genes was too high for them to be included. So it was greater than 2 standard deviations. And we can then map onto these patients clinical diagnosis of the 5 different known subtypes and ask if it is stratifying the patients into the known subtypes. And what we find is when the expression of the 16 genes is low, you have predominantly normal tissue. But as it increases in expression, you have enrichment of mesenchymal subtype. And you can then use this signature to ask whether it is also telling you something about survival. So you can ask, do patients in the first fifth or the first quintile of this trajectory survive longer than patients in the last quintile? And so you convert this into a survival map, and then you can do that assessment, and you can find that, yes, these signatures can stratify patients into meaningful clinical outcomes. If you had information regarding drug responsiveness within these subgroups, you can use that information as well to then predict in the future if a patient falls into the signature, how likely they are to respond or not respond to a given drug. And then for each of the signatures, we have this map I mentioned earlier. We have a map of how mutations map to the regulators who control these biclusters of genes that are associated with biological processes associated with the hallmark of cancer. So the relationship between the transcription factor and microRNA and the regulation of the bicluster is supported by mechanistic evidence, because we know the location in the genome where they physically bind to control. So that's mechanistic inference. The association of mutations to expression of regulators and genes in biclusters is a causal inference. So there you can say patients who have this mutation have high level of expression of these genes, but you might not necessarily know the mechanism by which that the expression is increased. And this is the network. So you can convert all the piles of data into a complex but still simpler network than the data themselves. And it allows you to then probe this information for specific hypotheses. For example, you can ask, what is the consequence of having mutations in NF1 and PIK3CA, which are predicted to control IRF1 and increase its expression, which is regulating genes associated with lymphocyte invasion. So what is the consequence of this mutation in patients? And this network predicts that the tumors will have increased lymphocyte infiltration. We can go and test that. We can do a series of tests. I won't go into the details, but for every edge predicted in that network, we can do independent validations to confirm that if you have a mutation in a tumor, you do see increased level of IRF1 expression. You do see increased level of expression of bicluster genes. And if you remove the IRF1 expression, you lose the correlation between mutation and expression of genes associated with the cancer process. So a series of tests you can run before going a little deeper into some phenotypic validation. And because the network predicts that 12 of the 27 genes are associated with MHC class 1 antigen processing and presentation machinery, or APM, you can predict that when you have the mutation, this pathway will be overexpressed, and as a result, you will have increased tumor lymphocyte infiltration. And there was an independent group that did a study on this, and they found that indeed, when the mutations were there, you had moderate to high lymphocyte infiltration in tumors, as opposed to patients who did not have these mutations. Now, we can also use the network for doing drug discovery. And because we know the topology, we can find drugs or interventions that hit nodes that are connected in characteristic ways to predict synergy. And something Lee was mentioning, we want to find double and triple drugs that can be used against specific tumors that have certain characteristics. And this is one way potentially to do that. I'll skip all of this, but essentially there's a lot of validations that we did, genome-wide screens of knockouts of transcription factors, targeted knockdowns of transcription factors and microRNAs overexpression and so forth. And for each, we measured the consequence on apoptosis or proliferation to confirm that the network was making accurate predictions. So we can then do things like this, where we can, as I mentioned earlier, we can map a new patient onto a trajectory and potentially predict a clinical outcome based on characteristics of patients that grouped into that subgroup. And because we know the network underlying that signature, we can then, in addition to predicting clinical outcome, we can predict what the dysfunction is. And based on that, predict drugs that could be used that have been used in GBM or even repurposed drugs from other indications, because the targets of those drugs are in the signature that is associated with some clinical outcome and some hallmark of cancer. And you can combine the drugs or drugs with anti-microRNAs or microRNA mimics or siRNAs against transcription factors. And there are new technologies coming online to deliver these as therapies. And when you have certain topologies, you get synergy. And so for a given patient, you can make a prediction that this patient is most likely going to respond to this drug and this microRNA mimic, and we need to run this specific test on this patient. And when you run these tests otherwise on a broad cohort of patients with mixed characteristics, your responsiveness data will be not as clean. But here, if you choose the patients based on these characteristics, the likelihood of finding a responsive treatment is potentially higher. And we showed that here in a glioma stem-like cell line and compared it against a neuronal normal cell line and showed that the effect of the drug could be enhanced by adding a second agent against a microRNA. And the drug we used here is a pan-HDAC inhibitor called romidepsin that is made by a cell gene. It is effective but very toxic. So the question is, can we lower the dosage of the drug and then increase its efficacy by tuning in some other agent? So here we show that we lower the drug dosage, and you see the proliferation effect going down to almost being negligible. And then you tune in the microRNA mimic, and you start recovering the effect of the drug again. So by combining this, we can now take a pan-HDAC inhibitor and potentially also make it specific, because the microRNA is specifically targeting the transcript of HDAC5. Right? So you can begin to take a generalized drug and make it more specific to a specific dysfunction in a patient. So I screamed through this, but there's a paper that you can read that describes the details of this approach. And there's many additional papers that describe the algorithms that are cited in this paper. And the work was done by a group of people, but Chris Plaisir was this postdoc in my lab and now a senior scientist who's done this amazing job of developing and integrating these algorithms. Just wanted to point out that this approach is a generalizable approach because we use the same approach to address drug resistance in TB. I won't go into the details of what we did, but essentially what we found was there's a drug that FDA approved, and this is the first drug in the last 40 years that has been approved against TB. And the drug resistance issue in TB is getting exacerbated, so we need new lines of drugs. But this drug has a delayed killing effect. And we found out using this approach that when you treat TB with this new drug, there's a network that is triggered that can induce processes that allows TB to tolerate the drug. So we found a second drug that disrupts this tolerance network. When we combine the two drugs, we render TB even more sensitive and the delayed effect is gone. Right? The same conceptual approach could work in cancers, but here the challenges are in matching these screens to patients-derived xenografts or cell lines so that you can run these screens in a meaningful way. So here's a summary. I covered it already. So I'll stop there, and I think it's the panel discussion now.
Yes. Thank you.
Thanks.